Hello, everyone. We'll give it a few more seconds before we get started. All right. Good morning, good afternoon, and good evening to viewers around the world, and welcome to our 4th annual Global Reaxys User Day. My name is Randall Kenyon, and I lead the Life Science Solutions team for North and South America at Elsevier. I have the pleasure of being your host today, and it's great to see you, our chemistry community, together here online. This event is recorded live, and we have a great lineup for you today. All are experts, and they will present a variety of interesting chemistry-related cases. Before we get started, a few housekeeping items. First, we will make all of these recordings available after the session so you can rewatch or view a presentation that you may have missed. Second, there's a chat function that you can open at the bottom right of the screen that will show you the Q&A menu. We encourage you to ask questions during the sessions. You can easily click from chat to Q&A to polls to handouts. In the handouts section, you can download additional resources. Following each set of presentations, there will also be a survey available for you to answer. Please enter your questions in the Q&A tab so that we can see and answer all of them. If we don't answer a question live, we will answer it after this event. To access closed captioning, click on the CC icon and then select your preferred display language. Finally, please do share your feedback so that we can continually improve the event. Now, it's my pleasure to introduce our first speakers, Dr. Oleksandr Grygorenko and Dr. Ilgin Demirer. Oleksandr shares his time between his alma mater as head of organic chemistry department and leading researcher at Shevchenko National University of Kyiv, and also as a consulting scientist at Enamine. Oleksandr received his PhD in 2007 and Dr. Sai in 2019 in organic chemistry. Oleksandr is the co-author of over an impressive 250 papers and 8 monograph chapters. His scientific interests focus on organic and medicinal chemistry, including lead-oriented synthesis, conformational restriction, organoboron, fluorine, and sulfur chemistry, and non-proteinogenic amino acids. Dr. Ilgin Demirer is a customer consultant at Elsevier, where she specializes in supporting R&D scientists and engineers across Eastern Europe, Germany, Australia, and Switzerland. Ilgin holds a PhD in chemistry from the University of Saarland, complemented by a master's degree in chemistry from Freie Universität Berlin in Germany. Throughout her studies, Ilgin has actively contributed to numerous projects focused on main group and coordination chemistry, as well as organic synthesis. Today, Oleksandr will share insights from exciting work in a talk titled Pushing the Boundaries of Organic Chemistry, Transforming Concepts into Molecules. Please welcome Oleksandr and Ilgin to the stage. Thank you.
Thank you so much for a very kind introduction and for inviting me and having me here today. And I would like to start with a few slides about our company, about Enamine. I think that many of you know about Enamine. It's a scientifically driven company which provides a number of chemical and recently biological services. It is privately owned and founded more than 30 years ago. The main facilities are located in Kyiv, Ukraine. And now we have over 1,000 synthetic chemists and several important assets that help life sciences and in particular drug discovery to do their research. In particular, our main asset is, which I will be talking about, is a huge collection of building blocks for various organic and medicinal chemistry synthesis. This is a significant part of the world's commercially available stock, and it is used to build all other our products and services. We also have the world's largest commercially available collection of stock screening compounds. This is also this is achieved also because we have a lot of different building blocks to synthesize them, but also because we developed through years efficient methodologies to do that. We provide a number of chemical services and also started to develop biological services business, and it's going quite successful. The biological team of Enamine called Bienta, and you can do integrated early drug discovery research from there. We do not do all our own drug discovery research. We are fully services oriented, and also we have a number of representative offices in throughout European Union, in Germany, in Riga, and also in US, Japan, and China. Our main asset is a collection of building blocks, which was constantly growing through recent years. We increased it more than 6 times, even more than through the last 15 years. We add around 2,000 new building blocks each month now, and all of them are synthesized in-house and developed and designed also in-house. We focus on chemotypes that are attractive for medicinal chemistry, first of all, of course, but there are some specific cases where we provide building blocks for completely different areas, for example, material science and something like that. And we have a huge diversity of chemotypes there in all terms, in particular in terms of functional groups. We have quite an impressive collection of most common reagents, carboxylic acids, primary amines, secondary amines, and etc. And also we have some more exotic compounds that are not so well represented at the market. We have quite good representation there. These building blocks are all compatible, or most of them are compatible with parallel conditions, synthesis conditions, and they are used to construct our screening collection, both virtual and in stock. And of course, all our building blocks are accessible through Reaxys, and we are very happy about that. And recently, we have signed an agreement with Reaxys and added our make-on-demand building block collections. It's more than 40 million of these compounds that can be synthesized very easily in a few steps, and so from available to us reagents. So they can be considered as really accessible. And these huge collections can be found at Reaxys, through Reaxys, and ordered from Enamine. Upon expanding our collections, we use 2 general approaches. They are quite straightforward, I think, but nevertheless, we either try to introduce some new and interesting chemotypes that have been underrepresented in both scientific literature and commercial collections, and or were not presented there at all. And also we try to develop some synthetic methodologies that allow us to cover wider areas of chemical space. And I will show you 2 examples today which demonstrate each of the approaches, and were published recently by our scientists. So the first case will showcase fluorinated oxetane chemistry, and many of you might know that oxetanes are quite popular, have become quite popular in drug discovery recently, although they have been known in drug discovery and medicinal chemistry for quite a while. For example, oxetane ring is present in the well-known anti-cancer agent Taxol or paclitaxel. But significant interest to oxetanes increased in the middle of 2000s, and after their publication by Eric Carreira and von Laroche chemists, where they demonstrated isosterism of oxetane, bioisosterism with several important structural moieties, again dimethyl moiety and carbonyl group. And despite this increased interest, there are a lot of synthetic challenges in preparation of oxetanes. We have put a lot of efforts into developing synthetic methodologies for providing oxetane-containing building blocks. But the story I would like to show you is related to the specific case of oxetanes, that is fluorinated oxetanes. These oxetanes are quite largely underrepresented in the literature, and they are quite interesting because many of synthetic drugs now contain fluorine atoms, and novel fluorinated motifs are needed to expand the chemical space for biological studies efficiency. So in this story, we introduce this difluoroxetane moiety, difluoroxetane group, and perform its thorough characterization by physicochemical management, some theoretical studies, and finally we performed some biological evaluation of bioassociatorism using this group by preparing analog of a known sirtuin inhibitor. So how we started? We started with looking the literature methods towards such compounds. And of course, we use Reaxys extensively for this purpose. And what we found through Reaxys is that, in fact, this difluoroxetane moiety was mentioned in the literature, but only in a few works, and no building blocks, no compounds with functional groups were available at that moment. And the synthetic scheme that was proposed by one of the authors relied on quite interesting chemistry. That is, that includes rearrangement of difluorinated cyclopropane derivatives into through ring opening into this interesting bifunctional intermediate bromoketone, and then cyclization of the formamide. There was only one example without any functional groups in the literature. So our first task was to implement this method, implement this approach into the synthesis of building blocks on multi-gram scale, so that they will be available to medicinal chemistry community on large scale. And we achieved that using the scheme shown on this slide, and introduced several quite nice side functional groups, including secondary amines, for example, or aryl halide, and even performed some transformation to obtain carboxylic acid, for example. So indeed, we demonstrated that this approach works well on at least the decagram scale, and these compounds can be accessible and now can be introduced and incorporated in the molecule of interest, even very easily. But we didn't stop on that and started to look at the physicochemical properties of the synthesized compounds. We started with pK determination of the corresponding model derivatives. Apart from difluoroxetane, we also selected a series of other substituents which we think that can be a target for replacement with this new moiety. And it was found that, as might be expected, difluoroxetane slightly decreases pKa of both piperidine derivatives when introduced at force position, and the corresponding benzoic acids when introduced at para position. So it acts as a weak acceptor. So it is much more weaker than, for example, ester or acetal moiety, which we envisage that they can be potential targets for replacement. But also, of course, it is a stronger acceptor than, for example, simple alkyl groups that can be another type of side chains that can be changed by this group, that can be replaced by this group. In terms of lipophilicity, we have found that introducing difluoroxetane moiety into the molecule of interest instead of hydrogen can expectedly increase the lipophilicity. The delta log P upon introduction to the substituent is 1.6, which is somewhat higher than 0.5. That is somewhat higher than for non-fluorinated oxetane. So in this case, fluorine has increasing effect on the lipophilicity, but at the same time, the group is much more hydrophilic than most simple alkyl or cycloalkyl chains that can be targeted by biosteric replacements with this group. So one of the possible applications for this substituent is to reduce the compound's lipophilicity. We also performed more precise measurements of electronic properties for this group and performed classical Hammett constant determination experiments using kinetic experiments. And we have found that the Hammett constant value for this substituent is quite similar to that of chlorine. It's the closest one in this series of common substituents. And of course, again, it's intermediate between non-donor alkyl groups and more polar groups like acetal or ester that can be also targets for the substitution with this group. And we also performed stability studies for the model derivative 3E using a couple of standard reagents that are used in organic synthesis. And we have found that the difluoroxetane moiety is stable to a number of conditions, including, for example, simple heating at the air, hydrogenation, action of trifluoroacetic acid at room temperature, some standard oxidants. It is also stable at room temperature towards alkali and hydrogen chloride, but it is undergoes ring opening upon elevated temperatures. The limitation of difluoroxetane moiety included also strong reducing hydride reagents like lithium aluminum hydride. In this case, the ring also could be opened upon elevated temperature. Also, we were pleased to find that after storage for more than 5 years now at room temperature, this compound also remained unchanged according to LCMS and NMR analysis. So this group can be considered indeed as quite stable at the laboratory. And finally, we have, in collaboration with our biology services, Bienta, performed the study of tenovin-6 analog. Tenovin-6 is known inhibitor of sirtuin and is quite potent. So we replaced third butyl group with difluoroxetane moiety. In fact, we also used extensively Reaxys at this stage of the study because Reaxys has very convenient way to find biological activity information in the corresponding for the corresponding targets, and we just looked at the chemotype that we needed. In this case, it was parasubstituted terbutyl phenyl ring, and easily filtered compound that have reasonable or quite high biological activity. And when you retrieve the list of these compounds, it is a very nice feature that you can sort them. They are automatically sorted by the number of occurrences in the literature. So the most well-studied compounds like clinical candidates, for example, or even marketed drugs are founded at the top of the list. That is the way that we identified this compound. It's quite easy to do. And then we performed the synthesis of the tenovin-6 and its analog, and performed some measurements of biological properties and physical-chemical properties. So we found that in this case, indeed, the log P and log D values are decreased. So the first purpose of the isosteric replacement was achieved. The solubility at the same time was not affected significantly. Perhaps the most nice part of this was that the efflux was reduced and the clearance was already quite low for tenovin-6, but it was even lower for the difox analog. Activity was somewhat lower, but still the compound was active. So we believe that biosynthesis reason can be considered as proved here. So the fluoroxetane group, we believe that it might be quite interesting for studies, and we added several building blocks containing these site functionalities at the site chain group to our building block collection after this study. And the second part of my talk, I would like to to present to you is related to CC coupling reaction using organoboronates. Of course, we all know organoboronates for their capability to undergo palladium catalyzed couplings, for example, Chen-Lam couplings and other similar reactions. But new direction for this chemistry is their radical reactions, and there are a number of transformations with this in this way, in this view, so we can achieve introduction of radical species by addition to double bonds. So in this case, we retain the boron substituting group, or we can replace it through addition elimination, some kind of addition elimination. And there are a number of functional groups that have been introduced through this approach, including some interesting fluorinated substituents. And there are some modification of this reaction that undergo through the organocopper intermediate formation, and then also involving radical chemistry. And we thought about this a lot and tried to extend this to various side chains, especially those containing fluorine atoms. And we turned our attention to this reagent number 1, which is commercially available difluoromethylene source, and it's quite accessible compound. And in fact, there were some examples of attempted CC coupling with this reagent using alkynyl boronic derivatives, and the known approach included, in fact, copper salts that we were thinking about. But in this reported case, the addition occurred to the double bond, so the corresponding boropenicolates retained in the molecule. In our work, we tried to achieve the CC coupling product, the formal CC coupling product, and we tried to avoid some expensive or complex catalysts. And again, we looked in the literature for the transformations of this compound number 1. We used again Reaxys for that. And we have found quite nice work published in Organic Process Research and Developments in 2018 that include a coupling of compound 1 with halopyridines that involved copper catalysis, copper metal, which is quite convenient, quite usual, very cheap, very old catalysts. So we tried to study this, apply to these conditions to our reactions, and although the product was initially formed, the yield was not too good at first. So we performed some optimization, and it was found that we can add the ligand for copper stabilization in solution and reduce the amount of catalyst to about 1.5 equivalents, and still have quite reasonable yields of the product, especially after additional optimization of the reaction time. And these optimized conditions we have used to we have applied them to a series of other compounds. So the scope of the method was quite nice. It included various dye-substituted derivatives in having the acyclic, heterocyclic substituents groups with additional functional groups like ether, for example, or protected amine, acetal and nitro groups, and so on. So some other representatives included monosubstituted derivatives, and there were some unusual regularities on stereoselectivity of the reaction, in particular typically with simple alkyl groups with functionalized side chains. The stereoselectivity was quite low. We had nearly 1-to-1 mixtures of E and Z isomers, but with aromatic derivatives, the stereoselectivity was quite nice, and we had only E isomer in this case. Maybe this is related to the stability of the corresponding radical species or perhaps to some steric factors can play in this case. But nevertheless, we didn't study this too much, but nevertheless, the method worked quite well for quite large range of site functionalities. And we also applied these methodologies to some natural products, for example, to camphor, astron, and vanillin, and it worked well with these natural compounds. So the method can be even considered for some late-stage functionalization if that is necessary. We tried some other esters at this moiety because with this particular substrates, the ethyl ester was quite volatile. It was difficult to isolate, but when we replaced with stat-butyl group, it worked quite well. The only exception we have found that didn't work well in our case was included boronates with aryl group at the alpha position. In this case, the addition product was formed at the main product of the reaction. With these results in hand, we tried to implement this into building block production again, because we demonstrated that the reaction is has quite good scalability. So this simplest representative was prepared at almost 80 gram scale from a single run, and we can perform some typical transformation to transform ester moiety to functional groups that can be useful as potential building blocks. So the corresponding carboxylic acid was isolated as a potassium salt. It is not very stable in the form of free acid, so we store it as a salt. The amide could be obtained quite easily. Oxidation is possible, reduction to corresponding primary alcohol, and finally hydrogenation of double bond with retention of the remaining part of the molecule intact. And we also thought about the mechanism of this reaction. So based on the literature data as well as some additional experiments, we assume that the reaction starts with formation of some copper species that undergo reaction with compound 1, forming the corresponding copper 1 complex and the free radical 12. That is one of the key components in the key intermediate in this reaction scheme. Then transmetallation occurs with alkenyl boronate, and organocopper intermediate can undergo recombination with this radical species, either by radical addition to the copper containing site, or by attack of compound 12 at the copper and with formation of copper 2 intermediate. In both cases, the product can transform into the target product either through reductive elimination or by removal of copper, reductive removal of copper, and regeneration of copper. We assume that the reaction is catalytic on copper because we observed formation of copper mirror on the reaction vessel. This is a photograph of the steering bar from the reaction mixture. So the copper participates in reaction and then reforms again. This is, of course, not the strongest evidence of that, but other evidences also support these results. And again, these results, this hypothesis relied to some extent on the use of Reaxys, because in this case we used search using some keywords about mechanistic transformation of alkenyl-boronic derivatives in radical reactions, as well as standard substructure search for similar transformations. With this, I would like to thank to a lot of people that were involved in this research, and especially for the difluoroxetane part, it's Oleksandr Leshuk, Konstantin Melnikov, and our biology team, analytical chemists, the people from physical chemistry department at our university, And for the baronate part, to Oleksandr Grygorenko and his group, who did all the synthetic work and mechanistic studies. I'd like to also thank to the founder of Enamine, the Professor Andriy Tolmachov, who was inspiring and supporting all this work, and to Enamine team that provided funding for this research, and also to Armed Forces of Ukraine, who made this research at these times possible for us. And thank you for your kind attention. And with that, I would like to give time to give the stage to Ilgin Demirer, who will be from Elsevier, who will be continuing the our talk with her part. Thank you. Ilgin, sorry, I think you are muted.
Thank you very much, Oleksandr. Sorry, everyone. So I would like to welcome you all, and thank you, Oleksandr, for the kind introduction and for the inspiring talk. Can you see my screen? Yes, I think. Okay, great. So for my part of the talk, I would like to demonstrate to you all how we can utilize Reaxys to access and analyze bioactivity data for compounds against a specific target. So for that, I will use the quick search, and I will combine the two sections that we have for the text-based search and our structure editor. And in our text-based search tool, I would like to specify our target. So for today, I selected an example. So as I type in, we can see that Reaxys already gives me some suggestions, and I will select from the list. And I will also go to the structure editor to define what I would like to have in my structure. So I'm not going to create a very elaborated structure, but I would like to specify that I want to have 5-membered or 6-membered rings which contains oxygen. So I will just add also repeating units, so we can include also 5-membered and 6-membered ones. And we can also say, I would like to have a trifluoromethyl group somewhere in my compounds, and we can just leave it as it is. Of course, you can be a bit more specific than I was. This is completely up to you and what you would like to have. So I will also select as substructure, and then now we can combine these two sections, and then we can click on find to see what we can find in Reaxys. So now we're in our preview page where we can see different set of results that we get. But since we're interested in the bioactivity of these certain substances against the specific target that we selected, I will go to the substances. So I click on view results to get here. So here we can see our results as a list. So we can also change how we sort our results. So therefore, you will find this option here, sort by number of references, but you also will have different options here. So you can just select the one that you would like to have. So here on the left-hand side, we can see our structures. Of course, if you would like to zoom in, you can also click here on the magnifying glass. And next to it, we will find different tabs. So we have various information for these substances that we get. But in this case, we're interested in bioactivity data against the target that we selected. So maybe it took already your attention, but we have this section called bioactivity hit data. So if I click on it, it will present me the specific data that I'm interested in. So we can find our target, and then we can also find additional details related to it. So for example, we have our px value, we have our unit, action on target, and of course the documents where these data points excerpted from. So these px values are generated in Reaxys, so we can compare our results a bit more easily. So depending on the px value, it shows us whether we have high or low activity. So we can also compare between those substances. So in this case, we can see that we got 548 substances. And if you would like to further customize our results, we can always use the filters that we have on the left-hand side. So in this section, we also have different options that we can use. So for example, you can select depending on certain px values, or if you're also dealing with multiple targets, you can also select it under targets, or you can also select whether you would like to have, for example, commercially available substances. You can apply these filters to customize your results a bit more. Okay, we can view our data, but what if we would like to compare the substances? So in that case, we can find this bioactivity visualization tool in the top right corner. So we can click on it, and then it will open us this heat map that we have. So we can see that we can find our target as well as our substances on the y-axis. So if you're also dealing with multiple targets, you will also find multiple targets on the x-axis, but so far you can see it like this. So in this section, we can see some values, and these values stand for the px values that are generated in Reaxys. And if you would like to sort your results, you can always click on this 3 dots, and then we find sort by activity. And then now we can see it goes from highest to the lowest one. So of course, if you would like to view our data points as well, so I can click on this px value, and then it will open this page here by activity detail, and then we can also view the details here. So as we saw previously. And if you would like to also change the settings, so on the top part, you will find the settings button. And in this case, we can find this heat map or bioactivity visualization. So on the x-axis, we had targets, but you can also find multiple options here. So you can just customize it according to your needs. And in the y-axis, you can also do the same. So here also you can select what you would like to view. So in this case, it's maximum of px, but you can also change it in this drop-down menu. It is also possible to export our results. So for that, we will find on the top part this export button, and then we can click on it. And then we have also several formats that we can select. So we have our new layout as well as other options if you would like to use. And we can also change what we would like to export. So in this case, by default, it's all available data are selected. But if you're interested in a certain data, you can always change it in this settings part and export your results. So since we mentioned about the filters, you can also apply more filters if you like while you're on the heat map. So in order to do that, I will click on this arrow, and we can see here multiple filters. So for example, if we say I'm interested in certain action on target, we can also select it here. And while we're on the heat map, we can also customize it a bit more. That was all from my side. I would like to thank you all. And if you have any questions, please let me know in the chat. I will be happy to answer.
Thank you, Ilgin and Oleksandr, for the wonderful presentations. We have time for a few questions. The first question is for you, Oleksandr. How do you stay on top of what the trendy new medchem attractive compounds are and make sure your library contains them?
Okay, so that's quite a good question because, yeah, there are several approaches that we are trying to do at Enamine with that. So the main idea is that our chemists and our production managers and even our CEO tries to put a lot of efforts into standing in time with current chemical literature, of course, and being updated on that. And our chemists generate new ideas constantly, and we have regular meetings at Enamine that perform some evaluation of what has been proposed by them, and then it is put into production. The most interesting thing are put after discussion are put into production. So this is constantly undergoing changes. And recently we have also tried to put a lot of efforts on even predicting the upcoming trends. It is the most difficult task to do, of course, but we think that we are more or less successful in that way. So we try to generate structures that can be interesting for future chemists. So the main advice in this area is, of course, to stay with trends that are published in literature, in the recent literature, and stay in touch with medicinal chemistry community on that.
Sir. Another question for you, Oleksandr. Your team analyzed and shared a number of physical ADME and Lipinski-Verber properties for these substituted compounds. In your experience, which one of these properties are most valued or important for drug discovery efforts?
Well, in fact, it's quite a difficult question to ask for me because, as I mentioned, we do not do our own drug discovery research at Enamine, but of course we have some feedback. And I think that we try to analyze those properties that should be most important for medicinal potential medicinal application in medicinal chemistry. I think that lipophilicity and metabolic stability might be that properties.
The next one is for Ilgin. Is it possible to automate or save filter settings for recurring searches?
In a way, we can do it. So I can share my screen again just to show maybe. So maybe I can use the same so in this case, we had our target and our structures. So what we can do here is also we can go to the query builder. So in the last part, maybe you remember, like I also mentioned that we can add details such as a substance action on target. And then here in this field, you can also add more details in Query Builder to create a bit more elaborated search queries. So what we can do here is also we can add more details such as substance action on target or certain px values, so you can combine all these sections. And afterwards, if you would like to save them, what we can do here is like we have this save button. We can either save it as query or form. So if we choose query, it will save it as a JSON file, and afterwards it will be downloaded in your computer, and you can import and further edit, or you can just search as it is in Reaxys. But we can also save it as a form, and there are two options here. So what you can do here is you can either save it as empty fields. So if you're using a certain combination of fields quite often, you can just choose the empty fields, and afterwards every time you can just fill it according to your needs. Or you can also include its field version. So I think I have an example that I can share maybe. So here, for example, I saved it, and then we can see the saved version, and afterwards I can again search it in substances. I hope it was helpful.
Indeed it was, Ilgin. Thank you. With that, Oleksandr, Ilgin, thank you for your presentations. They were quite insightful. To our audience, the next presentation will start in about 20 to 30 seconds, so please hold tight. And if you see a survey, we'd love for you to short out the short questionnaire so that we can gather your feedback. Thank you very much.